In this video, we will be learning about the transcription process in prokaryotes. Put simply, transcription is the process of transcribing a DNA sequence into an RNA sequence. Now, transcription has three main stages: template binding and initiation, elongation, and termination. In the first stage of template binding and initiation, the promoter region of the DNA is recognized by the RNA polymerase enzyme. This is done by the sigma factor of the RNA polymerase, which converts the RNA polymerase from a core enzyme into a holoenzyme. The holoenzyme recognizes the -10 consensus sequence, commonly known as the TATA box, and the -35 consensus sequence in the promoter region. These boxes are extremely important in order for proper binding to occur. For example, if the -10 box is deleted in a gene that codes for an enzyme to metabolize sugar, the holoenzyme would not recognize where the promoter region was, in which case transcription would never occur. A not-so-sweet ending. Once bound, the RNA polymerase holoenzyme separates the DNA strand, providing a template strand and a coding strand, making a transcription bubble. Now that the RNA polymerase has found the promoter and begun the process, the sigma factor leaves, allowing for stage 2 to begin. In stage 2, chain elongation occurs. To do this, the RNA polymerase needs a template strand to use as a template, hence the name, in order to build a complementary RNA molecule, which is why a primer is not needed. Something to remember is that the RNA is often written 5' to 3', and nucleotide bases are always added to the 3' end of the RNA. With this in mind and the direction of the transcription given, we can distinguish that the bottom strand is the template strand, while the top strand is the coding strand. The coding strand will have the same nucleotide sequence as the RNA strand, except that the uracil takes the place of thymine in the RNA strand. These bases are added to the RNA by cleaving two phosphate groups from RNTP from the phosphodiester bonds. The last stage of transcription is termination, which can occur in two different ways: row-dependent and row-independent. In the row-dependent termination, a protein called the row factor binds to the RNA sequence and moves towards the 3' end of the RNA. Once the row factor reaches the 3' end of the RNA, it uses a helicase enzyme to separate or unwind the DNA in the RNA's connection, causing termination. In row-independent termination, inverted repeats are present in the DNA sequence, which causes the RNA strand to form a hairpin structure. This structure inhibits the RNA polymerase, causing it to pause. Directly after the inverted repeats, the string of adenines in the DNA strand, which in turn causes uracil to form in the RNA strand. These AU bonds between the DNA and RNA strands are very weak bonds, promoting dissociation between the DNA and RNA pairs, in turn causing termination. These are the references.